Good morning, everybody. Thank you very much for coming. This is an initiative of our center, which I will talk about a little bit, and also about an initiative that was set forth by Sergio Pantano and friends a couple of years ago, which is the SAIMS, the South American Initiative on Molecular Simulation, which is that congregates South American investigators in molecular simulation, especially in biomolecular systems. And we are very excited that this is actually taking off, and we hope to, reach cruise height and actually fly off. I will spend, the meeting is very exciting. You have several very nice talks on different subjects, and also hands-on courses, which we hope will work well. And at first I already apologize for some of the misorganization that is entirely due to my fault. I want to spend just a couple of slides just to present to you the center that we are running here at the University of Campinas. This is one of these research, innovation, and dissemination centers funded by FAPESP. FAPESP is the São Paulo State Research Foundation. It's a very important funding agency in Brazil, but it only funds research in the state of São Paulo due to São Paulo State contributors, tax contributors. And there are 17 of these new centers, and these centers are expected to carry high-level research, technology transfer activities, and also dissemination and science education activities. So our center is named the Center for Computational Engineering and Sciences. The name sometimes causes some confusion because people think that we are engineers. And I say, yes, we are, but at an atomistic level, or molecular level, if you will. But the center is, the idea is that we hope to establish a multidisciplinary center, and the center is dedicated to the development and application of high performance and data-intensive methods in a variety of computational problems, such as molecular sciences like ourselves, and the physicists. There are several very high-level physics going on in this center. Mechanical engineering, computational mechanical engineering. There is also research in bioinformatics, computational biophysics, and of course computer science, which unites us all. And the idea is that not only research, but we're able to provide through this funding, provide the necessary infrastructure to carry out challenging research. That's the idea that we have. So the center has faculty usually from Unicamp, computer scientists, physicists, chemists. Leandro Martinez is here. Mechanical engineering, two friends, these ones are there. One of them is not in the country right now. And there's also biology. And this biology part here, there's also a connection with one industry in the bioethanol sector, which is already participating. And the applied mathematicians, and also some international associates, Dario Estrin being one of them. He will arrive the day after tomorrow, or tomorrow, the day after tomorrow. So the idea, the underlying concept is that, you see, this is many disciplines involved: computational physics and chemistry, computational engineering. They work on finite element methods, boundary element methods, things related to the macrocosmos, right? Not macrocosmos, but macro materials, right? Yeah, macrocosmos from a molecular perspective. It's really. And the computational geophysicists, they work on actually analyzing seismic data and different areas. So what really what makes us join together here is the high performance and the modeling and the data infrastructure that we hope to be able to provide. So one of our challenges is to increase research in the boundaries so that we can do actually cross-disciplinary work. So this is the idea of the center. And we have fellowships sometimes and resources to carry this research. Things like, for instance, this workshop here that we have to be able to promote every once a year. So I was going to stop here, but I decided not to because there's a very nice work that one of my postdocs is doing. And he's not here today because he's going to spend, he's going to Holland. He was going to work for the competition, Steven Marink, right? And well, Marink offered him a 3-year fellowship, so he's going to stay there. And today is Mother's Day in Brazil. So we are really workaholics here. I actually forgot that was Mother's Day when I put the data. So he's not here. So since he's not going to present this, I wanted to present some of this work. And I know that I'm already over, the schedule, but I want to tell you a little bit about this work so that we hope to get also some input from you. So the thematics here, or the theme here, the topic that I want to tell here is about protein, ion, and gas collision cross-section. Why is this important? Because we are thinking that we are reinterpreting data, experimental data on ion mobility mass spectrometry. And this also helped, this provided us an opportunity to develop some high performance computing. So we are interacting with people, experimental people that do mass spectrometry data using ion, electron spray ionization and ion mobility experiments. So this work was developed by Paulo Souza and Leandro Zanotto, who is also our software engineer in the center. He also did a lot of work on the software for that. Okay, so the idea here is the following. Everybody knows about mass spectrometry. It's a technique where you generate ions, you accelerate ions, and you pass the ions through a magnetic set of fields, and you can separate the beam according to the mass-charge ratio. Okay. So the idea is to use this type of experiments. That's what the mass spectrometry people want to do. The ones that are trying to understand protein structure, they use this idea to analyze large macromolecules, such as proteins. Okay, so and the idea is that they can, with very little amount of unpurified protein, they can pass through this mass spectrometer and they can analyze the different types of structure. Well, they hope that they can analyze some sort of type of structure, different types of structure based on the charge. At least they can separate and identify different mass to charge ratio. So the idea here is that if they take one of these beams, separated according to the desired mass to charge ratio, they can send this beam through a gas chamber such that they can accelerate this gas chamber, and then this is called a drift tube. And the idea is that they can separate the pulses. There are two structures here. One is an extended structure, the other one is like a more folded one. And the idea is that the extended protein structures will have a larger cross-section, and they will take longer to travel through the drift tube because of the drag due to the friction due to the gas provided by the gas. Whereas more compact structures will fly through the tube at higher speed, and so the time of travel is smaller, and so they can look at the population and they can say, well, this high cross-section means lower time, smaller cross-section goes faster through the tube. So analyzing these different types of peaks, they hope to understand or to at least separate, well, this is structured, this is a more extended structure, this is unfolded, this is folded. And so that is the idea. Of course, these proteins here are charged. They have a net charge. Sometimes they have not just a single type of charge, but they have a charge distribution. Some of them may be zwitterionic ionic. Okay? So the idea here is, again, the same thing that I just mentioned. You have a drift tube. You apply an electric field here, and there's a gas there that's under certain temperature and pressure conditions. And then you go through you submit the ions here, the protein ions, and then you detect them here. And you're looking at the drift time, you are able to conclude the average collision cross-section. And this is the idea that I just mentioned. You have the friction due to the gas, you accelerate the ions, and then you can separate according to the size, shape, and interaction with the gas. So it's very important to interpret this data to be able to understand what is the cross-section. What is the value of the cross-section of the certain structure with the gas? And then once you have that, you can try to look at what is the structure that corresponds to that cross-section. Okay? So this is the idea. Okay? So in addition to that, they also use something called cross-linkers. Okay? So in addition to submitting these protein ions through this gas chamber, drift chamber, they can also use cross-linkers, which are small molecules with a certain length. And these molecules, they covalently bind usually to positive charges on the surface, usually lysines, but not just lysines. And they do different types of binding. They can have a strap conformation, which is like on a single helix, on a single substructure, 2 different points. They can be in a bridge conformation where they contact residues that are far apart in terms of sequence. Okay? And they can also be in a dead end conformation. So just one of the ends of the cross-linker is bound there. So they do this so that they can actually create some topological constraints. Leandro has done very nice work on that. I'm not sure if he's going to talk about that. He's not going to talk about that. But some very nice work that's entirely different from what we're doing, but it's a much harder problem. But he's younger and braver. So the idea here is so what they do is they use these cross-linkers so that they can actually, after they do the digestions, they can see which residues are close to the other residues because of this, right? So they don't know the structure, but they say, well, this residue is near that residue because under the digestion they came together. So with this, they separate different fragments. They digest the proteins, they separate different fragments, and then they try to reinterpret this data. They try to reinterpret, based on this data, they try to reinterpret, or not to reinterpret, but try to propose what type of structure. And so the idea here is that they have candidates, and they say, well, this candidate, can this be topologically connected? Yes, no, based on this one. So they can separate out which type of structure corresponds to no. Can someone? I'm feeling hot, are you? Yes. Você pode ligar lá de trás também, por favor? Okay. So this is the idea. So, but the question is, are proteins really stable in gas phase conditions? We don't know. What is the charge distribution under ion mobility mass spectrometry conditions? We don't know that. Okay, are these estimates for cross-section, how reliable they are? Okay, so this is where we try to come in. Okay, so as a proof of example, proof of concept, let's say, even for the experimentalists to try to understand the structure using this type of data, they use a ubiquitin, which is a very well-known structure. And so this is actually the cross-section distribution based on the time of flight, the experimentally determined. The black curves is the unmodified protein structure. You see there are two types of distributions. And then you have with the different types of cross-linkers. And I won't go into detail what these different types of cross-linkers are, but one of them preserves the charge, the other one does not preserve the charge, okay? The other one, when it binds to a lysine, actually drops one positive charge in the molecule. So it alters the charge state of the protein. So, but the idea, what they see here is that when they use cross-linkers, there is a shift to more compact structures. They want to try to understand why is that so. And our initial idea was, well, let's put this in water, let's put this in vacuum, let's compute the structure. We have a program that can compute the collisional cross-section and see what comes out. And what came out was really completely nonsense, way off the experimental data. Nothing actually converged. So this prompted Paulo to investigate better and try to mimic, try to reproduce the experimental conditions. Not just take protein in water, take protein in vacuum. We thought, well, protein in vacuum is going to denature that part, is going to extend this. And that's what experiments actually are interpreted. They say there's some helix, let's say, that's on the edge. They say, well, this one detaches and goes there. And this is a lot of hand-waving arguments that were never actually seen, not even in simulations. So, so we try to mimic what are the ideas behind experimental data. So the three main assumptions of the approach is that first you have electrospray ionization. This is in a water phase. They ionize this, and then they dehydrate at a higher temperature. And the protein goes through a certain ion guide, which actually accelerate the ion particles. And then it enters the drift tube, which is filled with some gas like nitrogen, sometimes now more recently carbon dioxide, but mostly helium. Usually, the former experiments, mostly helium. In the industrial machines, these are usually helium or not. And so this is what's actually going on in the experiments. So what we see here is that, well, ubiquitin is in a folded conformation. After dehydration, there are many different types of charges, charge distributions that then can place. The charge is +6 or +7. But you may have +6 how? It's 10 positive ions and 4 negative ions, or you can have 6 positive ions and 0 negative ions, and so on and so forth. And every charge distribution, not just the charge state, but the charge distribution, will contribute to the electrostatic interactions, and this will affect, or most likely will affect, the conformation of the protein and therefore the cross-section. Okay? So the other thing is the collision with the gas. Okay, so they apply an electric field, and these ions accelerate through the drift chamber, and then the drift chamber will collide with that, and that's how they measure the cross section, looking at the time delay along the travel time along the tube, right? Okay, and the expression that I presented before. So what Paulo did was to try to actually simulate this part here. So the first idea was he went there to the lab, looked at the apparatus, got the measurements. So how large is the electric field? What's the range of the electric field? How they applied? So he can, using the mass and the charge, he can compute what is the final velocity of the beam as it enters the gas, the drift chamber. Okay, so the protein with its charge, vacuum, enters the drift chamber with a certain velocity. He can compute the range of this velocity, known by the range of the velocities, because he can, he knows the electric field and the potential that is applied to that bit. So what he'd look at is that first. So initially, as the beam or the protein enters the drift chamber, it collides violently with the gas. And every collision increases the internal temperature of the protein. Okay? So as it increases the temperature of the protein, if the collision energy is sufficiently high, the protein structure is lost. You actually if it's sufficiently high, you can actually unfold the protein entirely. Okay. However, as it goes through this is in the initial stage, as it passes through the drift chamber, there is a thermalization due to collision of the gas. So the gas is maintained at a certain temperature, and every time a molecule hits the protein, it absorbs some of that energy. And what you see here, you see this is an actual a time history of the velocity of the protein. You can see that it suffers sudden drops. This is the center of mass translation velocity. It suffers certain drops, and these are due to very energetic collisions due to the gas, with the gas molecules. Okay. After several nanoseconds later, it cools down, and this here is what we call this collisional annealing. Okay, so it's a thermalization, it's a cooling down of the protein as you go temperature. So what he did is that at this part here, he looked at this structure. Okay, so what are the structure at this region here? Okay, after it has gone through the drift chamber. So this here takes very fast. So the actual measurement, time of flight, is taking place here. The protein, when the protein is traveling this section of the tube, it's already in this sort of regime here. So he changes, the temperature can be as high as some 500 degrees, and this will depend on the initial collision energy at the entrance of this tube. So with that, he can compute for every population, during the trajectory, every population, different types of, he does that many, thousands of simulations like that. Thousands of simulations like that, and compute the cross-section. Okay, and then he can have different cross-sections for different collision energies and different charge states. Okay, so he can compute that, and he can also compute native contacts and all the structural indicators of this protein. He can calculate and he can monitor. And then I'm not going to go into detail, but the average of on this section here of the trajectory, they use to compute all these properties. And the most important here is this collision cross-section. So here are the results for the cross-section in the absence of cross-linker, in the presence of different types of cross-linkers, and under different types of collision energy. You see that as you increase the collision energy, the peaks unfold into more substructures. And this is because it heats, it denatures, but as it cools down after during the annealing process, it may fold into different types of structures. Okay, so he can have, there are several types of this types of the peaks are different, and he can examine the structure corresponding to every peak in this distribution. So he can go back there and see, well, when this is the gas phase, when there's a low collision energy, what's going on, when there's just zwitterionic ions, let's say, and then you have that, and other types of collisions. So he can identify the structures with the different peaks. Okay, so he can actually not only compute the cross-section, but he can tell you, well, the structure of that peak corresponds to that sort of globular thing. And this happens in vacuum. So this is a misfolded thing, right? And it's misfolded because it's in vacuum. There's nothing else to it interact with. And so it folds whatever And here is where the charge plays an important role. Okay, or the charge distribution plays an important role. Okay, so he can not only compute the cross-section, but he can also look at the structure and see what the cross-sections correspond to. So, and he can examine not only the role of the charge distribution, but also the effect of the cross-linkers. So the cross-linker provides a mechanical restriction, fine, but the charge distribution creates different types of interactions with the gas, okay, because they charge different states. And that part of the paper I didn't read, so I don't think I can explain that. Oh, see, yes, the mechanical restrictions you're seeing here, the mechanical restrictions, depending, because there are many places where the cross-linkers can link to. And the experimentalists don't know. They know it's only a lysine, but they don't know where. There are many, many lysines. And there are many different possibilities for the cross-linking to bind to. So Paulo had to do different types, binding to different places, trying to understand it. So he did like, I don't know, tens of thousands of simulations like that. Okay. And, well, no. Sorry. It's missing the most important part. It's missing the most important part. I wanted to show, well, there's an important slide missing here. So embarrassing. Is the slide where we compare with the unmodified ubiquitin, the distribution of cross sections, where we compare this, where the experiments are. Yeah, we compare these things here, unmodified with different types of ligands. We compare this with the results that we obtain from here. So we don't know the populations. So you do some sort of a two-parameter fitting. Main peaks, the main contributors, it's a certain population of that, a certain population of that one, and you reproduce the experimental profile beautifully, okay, with a small shift here and there, because this experimental data is also very dirty. The experimental data is not dirty, but the road to it is kind of dirty, because you have to do lots of calibration and all sorts of things. But the match between the computed and the experimental data is beautiful. It could only be possible because of this ingenious, actually try to look at very carefully, try to reproduce the experiments. And the interpretation is also very important. So here, to finalize, because I'm way over the board, just to how we compute this collision cross-section. There's a code that does that, written in 1996. It's called the Modcal. It's very old code, very, very slow. Lots of go-to statements, right, Leandro? Really, it's an in-house thing, not ours. It's somebody else's, a famous program, actually. And the idea is the following. Here you have your macromolecule, and here's the colliding gas. So what you have to do is you can actually estimate the cross-section here very simply looking at the shadow of this. What is the average size of this molecule? And then you, just like projection of shadow, you can compute this cross-section. This is very naive. However, the interactions of the gas molecules with this macromolecule here is very different. Sometimes the gas molecule even hits and binds to certain pockets. So it's not just not computing the shadow. You actually have to get this molecule, collide with different velocities and different impact parameters, going here or going right head-on collision, different velocities of your gas phase. And not only that, you have to do this many, many times, but you also have to sample for orientation of your target molecule. Okay? Different orientations, hitting different velocities, different impact parameters, different configurations. Once you do that, you can compute this integral and you have the average cross-section. So the idea here is that not only take that into account, but also you take that for different conformations. You take this along the trajectory, right? So you have 50,000 conformations that you have to rotate to. So Paulo spent like 2 years doing that, the machine only for himself, one of the older clusters, and so when Leandro Zanotto came to our group as our code engineering or programmer engineering, he worked very closely with Paulo and tried to improve this program. So they rewrote the program entirely using C. And what they did is there are different methods. This is the trajectory method, which is the one that we are going to use. It's the most important one. And the idea is that they not only rewrote this code, but they make it parallel. They parallelize the code. Why? Because the idea, this is a very simple code to parallelize. I mean, it's not a very simple code to parallelize. The idea is very parallelizable, if you will, because you have different collisions, different orientations, so you can distribute this over the different processors, and you can do that. So this is working very, very well, and it's not in a CUDA version yet. Leandro is working to put that on a CUDA version. And more, we can actually do different gases, not just helium, because we can, so the protein you rotate, but the gas, you have to actually look at the different orientations of the gas. So this is very complicated. So what we want to do is, since it's a CO2, nitrogen, or something very simple, we want to use a Lennard-Jones plus embedded multipole models. So you have a dipole, you have a quadrupole, so that you only have to look at the trajectories regarding the center of mass, and the rotation of the solvent molecule, it's taken care of by the integration of the multipolar interaction, so that you don't have to deal with rotations of the. So the initial versions already dropped several times. You can do these calculations before. Every single calculation took like 3.5 hours. Now this, in a single node, takes only 6.5 minutes. So imagine now what can we do? We can take every protein in the data bank and run this and create a database of collisional cross-section, which is very important for people that are doing mass spectrometry experiments. They want to know what is this cross-section, what is this protein, what would be the cross-section, without having to do all that, without having to do all that, we can just get to a single point here for a given structure. Okay, so we can do that. And Leandro is working now on this to not only to perhaps extend this to every protein in the data bank, but inspired by Sergio Pantano's suggestion, we also have the PDB for the Zika virus. And it's hopeless trying to compute the cross-section for such a large macro object such as a virus using the earlier version of MobCal. But this one here can be, and this is a useful information for them, for many people in the area. They want to know what is the cross-section of this virus, of that virus. So we hope that we will be able to compute this in a reasonable amount of time using the new implementation. Okay? So, in conclusions, that's what I want to tell. We have a new code for computing cross-section, fine. But not only that, we have a new way of interpreting the ion mobility mass spectrometry data. And this is the most important part from the protein structure aspect. Okay? And I have to thank Fabio Goza from our institute. He's an excellent experimentalist on mass spectrometry, has a very nice lab. Alexandre Gomes, who did his postdoc with him, now is in this MS company called Waters. We hope we'll be able to sell them the code so that they can put the code, yeah, they can actually put the code into their packages of program, right? You can compute cross-section here. You just give me the PDB and, okay. And Alana dos Reis was a former student from Fabio's group. Okay, all right. And I have to thank Leandro, and I have to thank Paulo, who is not here, spending Mother's Day with Mom. Okay? All right. That's what I wanted to tell you. And thank you very much. Okay? Yes, Mauricio, por favor.
Oh, that was very nice. I have a question, maybe, I don't know very much about the experiment, but the charge state that you get is from the ionization of the residues, or those are from the ionizable residues, I mean the ones that take lose the salt?
No, they know the charge state for sure.
Of the whole system?
Yes, of the whole system. They don't know where the charges are, but they know exactly the mass to charge ratio.
And how do you do it to generate the configuration? Because there are lots of possible configurations.
Yes. They don't. The experimental papers, when you read them, they don't mention at all. They say the charge is +6. And they don't, there's a controversy, some issue saying, oh, is it zwitterionic or non-zwitterionic? Is the charge distribution important? They cannot usually address that. So in our case, we consider different types of charge distribution, where the residues are.
Yeah. But exactly that's the point. Do you consider on your charge sampling the ionizable residues, or you consider ionization of residues that are usually neutral, like for example tryptophan or
No. Don't know. I have to ask Paulo. But I think it's the ionizable. But yeah, it's not like it's not every residue. There are certain residues that are ionized. Yeah. Well, I hope I can answer the second one.
I've done cross-section calculations like ages ago.
Yes.
Small molecules.
Yes.
And obviously, small molecules have a very small integration range. What kind of integration range and grid do you have for it?
Oh, it's in the paper. I don't know, but it's huge. Also, we can do small molecules really fast. Really fast. We are actually trying to, this is, if we have a postdoc or somebody to do this, we can compute cross-sections for several types of molecules of interest to mass spectrometry. But this can be done really fast, much faster than mob call. Yeah? Okay. Sergio?
Really a huge amount of work, I mean, in terms of simulations, also work, not only computers wise, but also chem-wise.
Yes, a lot of phosphate.
So I wonder if you try different force fields, because at a certain point in your presentation,
yes,
proteins will unfold in vacuum, which is reasonably true. My question was whether the force fields will unfold the proteins, yes, because the force fields we have perhaps are not so good to work in vacuum.
Yes. Another thing that we are working is to implement the code to make it general to different types of force field. You just insert the right types of files, topology, force fields, and you can choose from force fields. The assumption that proteins will unfold in vacuum is they will perhaps lose some of the folding, but usually what we see is that they get more compact, not denatured, more compact. They have nothing to interact with except themselves. It's that. Okay. If no, one question, yes.
When the protein is flying on in the chamber,
yes,
one imagines that the protein will orient itself with the smaller cross section along the moving direction.
Yes.
That's taken into account in order to compute the comparative with the external data, or
if it's an extended object, if it is, it will come into the drift chamber like this, right? And as it collides, it will tend to align, right? Okay, and so that is taken into account when you do the calculations. You don't have to worry about the way it goes. If it aligns,
that's the cross section.
That's cross section that's going to be measured. Okay.